for giving me an opportunity to present in the brief webinar today. I'm Jisoo Ho, and then currently working in the University of Pennsylvania as the postdoc. Today, my presentation will be performed in English, but if you have any questions, please feel free to ask either Korean or English. So that today I'm going to present about the prefrontal regulation of the prefrontal cortical regulation of the REM sleep. REM sleep was first identified in humans around the 1950s. During the sleeping, researchers found that there is a period of the sleep with the rapid and then the jerky eye movement, similar to that the eye movement happens during the wake. Researchers also observed that the timing of this eye movement seems very closely related to that the dreaming, because that when the people are awakened from that when this eye movement happens, they usually report that they are dream, whereas that at the other point that they rarely report that they are dreaming. And then these periods of the sleep is defined as the rapid eye movement sleep, so called as the REM sleep. And as the study about this REM sleep proceeds, that the researchers also found that this sleep stage was characterized by the very distinctive brain activity represented by the EEG. The cortical EEG during the REM sleep shows the very fast and then the small oscillation, like the EEG is happening during the wake, which is very distinctive from that the large amplitude, slow oscillating EEG happens during the non-sleep. Later study also found that the REM sleep and its distinctive cortical activity is also observed in the other animals, like the cats or the rodents shown in here. And this distinctive EEG patterns during the REM sleep indicates that there is a very dynamic cortical activity changes happening, similar to that even the level of the activity happening during the wake state. Recent studies found that revealed more about how distinctive the cortical cortex behaves during the REM sleep, even sometimes different from that activity during the wake stage, despite of this similarity happening at the EEG level. The calcium imaging of the cortical neurons shows that the activity dynamics of the REM sleep is also expressed at the cell level. Parietal neurons of the cortex can be classified into the two groups, which shows that the opposite direction in activity changes during the REM sleep. The one group that shows the lower activity during the REM sleep compared to the wake or the non-sleep, whereas that the other group shows the highest activity during the REM sleep compared to that the wake or the non-sleep. This diversity in the activity of the cortex is also presented at the brain regional level, the larger level, brain regional level. The activity of the cortical regions, like the dorsolateral prefrontal cortex, is reduced, whereas that the other regions, including that the medial prefrontal cortex, and then the occipital cortex, becomes active, similar or the even higher than the activity happening during the wake. This association between the cortex and the REM sleep is also presented at the functional level. Deprivation of the REM sleep impairs the cognitive functions, especially related to the cortex, like the object recognition or the extinction of the conditioned fear. Also, processing of the learned information happening in the motor cortex at spine level also happens specifically during the REM sleep, not happening during the non-sleep. However, despite this close association between the cortex and the REM sleep, the role of the REM sleep in the regulation of the role of the cortex in the regulation of the REM sleep has not been given much attention. Previously, that the neural circuits regulating the REM sleep has been identified mostly in the posterior part of the brain, like the pontine area or the hypothalamic area, then it has been known that the REM sleep propagated start from the brainstem area and then propagated to the cortical area. So I wonder about the possibility of the regulation in the opposite direction, whether the cortex itself also has the ability to regulate the REM sleep. To study it, I target the medial prefrontal cortex, the MPFC area. Medial prefrontal cortex plays important roles in the various functions, including the cognitive functions, through its abundant connections with the other brain regions, including the brainstem area and then the hypothalamus area. Considering that the MPFC shows the functional impairment in the many psychiatric disorders, which are also accompanied the disruption of the REM sleep, I hypothesized that the MPFC would be an appropriate target for having an ability to control the brain state. So my first question is that whether the MPFC can regulate the brain state, especially the sleep state, and the basic event happening during the REM sleep. If so, and then which brain region can mediate this cortical control of the REM sleep, and then which regions would be the potential inputs for this regulation of the REM sleep. So first, to see how the activity of the MPFC neurons changes during sleep, I performed the calcium imaging of the MPFC pyramidal neurons using the fiber photometry. And as the animal enters the REM sleep, you can see that the activity of the medial prefrontal cortex pyramidal neurons increase significantly at the mice enters from the non-REM sleep to the REM sleep. So that the average activity during each state, MPFC pyramidal neurons shows the highest activity during the REM sleep, even more active than the wake state. And also at the state transition, especially at the non-REM to REM transition happens, that the MPFC activity start to increase even before the time point of the REM sleep onset, which means that which indicating the possibility that the activation of the MPFC can trigger the state transition, not the activity is changed after the state is changed. So to see whether the MPFC has an ability to induce the REM sleep, using the optogenetic stimulation, I activate the pyramidal neurons of the MPFC. And then when the laser is stimulated and then the MPFC neurons are activated, we can see that the strong theta oscillations start to appear on the EEG level, and then the mouse also loses its muscle tone, meaning that this mouse enters the REM sleep. Like in the video, when the laser stimulation was applied during the non-REM sleep, that the brain state of the animal changes from the non-REM sleep to the REM sleep. So which means that the optogenetic activation of the MPFC pyramidal neurons can induce the non-REM to REM sleep transition, means that the MPFC pyramidal neurons has an ability to regulate the brain state, and then they can induce the REM sleep. Then my next question is whether if the MPFC can trigger the non-REM to REM transition, then the MPFC is also involved in the maintaining of the REM sleep, not only just to induce the REM sleep. So to stimulate the neurons only during the REM sleep, I use the closed-loop stimulation system, detecting the REM sleep in the real time, and then we give the laser stimulation to only the half of the detected REM sleep bout, and then compare the REM sleep bout without the laser and then with the laser to see that the effect of the MPFC activation on the REM sleep duration. And the representative trace shows that the REM sleep episode with the laser stimulation is significantly longer than the REM sleep without the laser stimulation. This result means that the activation of the MPFC pyramidal neurons maintains the REM sleep state, so that the duration of the REM sleep, the average duration of the REM sleep bout is elongated compared to that the REM bout without the laser. Then the next, I tested that the effect of the MPFC inhibition on the REM sleep using the inhibitory opsins that the IC++. So that the bilateral inhibition of the medial prefrontal cortex during the REM sleep, showing that compared to the REM sleep without the optogenetic inhibition, that the REM sleep with the optogenetic inhibition seems to shows the reduced duration, means that the inhibition of the MPFC pyramidal neurons can inhibition of the MPFC pyramidal neurons that it shortens that shortens the duration of the REM sleep, and then it makes the mice wakes up from the REM sleep, which means that the MPFC pyramidal neurons can trigger or is involved in that the triggering and then maintaining the REM sleep. And the REM sleep is not the homogenic status. Along with this characteristic EEG and then the reduced muscle tone, as I mentioned earlier, REM sleep is actually first characterized by the rapid eye movement, to where does that the name of the REM sleep is coming from. And then this basic event of the REM sleep also happening at the electrophysiological level. During the REM sleep, there is a short period of the time when the LFP of the hippocampus shows the transient increase in the power, especially in that theta and then the gamma range, which is called as the basic theta, This basic theta event can be also detected on the EEG level, showing the higher peak frequency compared to that the tonic theta, the remaining period of the REM sleep. And when I compare that the timing of this basic event, that the eye movement during the REM sleep happens more when the basic theta happens compared to that the same length of the tonic theta during the REM sleep. So I wonder whether the MPFC pyramidal neurons can also affect the occurrence of this basic event happening during the REM sleep. To see that the role of the MPFC on this basic event during the REM sleep, I compare that the frequency of the eye movements and then the basic theta oscillation at the closed loop stimulation experiment in both activation and then the inhibition experiment. And the activation of the medial prefrontal cortical neurons increase both the frequency of the eye movement and then the frequency of the basic theta oscillation, whereas that the inhibition of the MPFC reduce the frequency of the eye movement and then the basic theta, which means that the MPFC is not only involved in that induce the REM sleep and then elongate the REM sleep, but also promotes that the more basic events happen during the REM sleep. And until now, I focused on that the role of the pyramidal neurons in the MPFC in the regulation of the REM sleep, which are the usually excitatory neurons. However, around the 40% of the neurons in the cortex is actually the inhibitory neurons. So I also wonder that what is the role of the inhibitory neurons in the medial prefrontal cortex on the regulation of the REM sleep and the basic event. So to target the inhibitory neurons in the MPFC, I'm using the VGAT cree mice, which is the representatory genetic marker for the inhibitory neurons. And using the channel of scene system, I optogenetically activated inhibitory neurons in the medial prefrontal cortex. And in contrast to that, the pyramidal neuron activation, that the activation of the inhibitory neurons in the MPFC suppress the REM sleep and makes the animal remaining in the non-REM sleep, so that impairs that the transitions from the non-REM to REM sleep happen. And the closed loop activation during the REM sleep, closed loop activation of these inhibitory neurons during the REM sleep also shortens the duration of the REM bout, and it can also significantly reduce the frequency of the basic theta, means that the MPFC neurons, unlike the pyramidal neurons in the MPFC, the inhibitory neurons in the MPFC prevent the initiation of the REM sleep, and then it makes an animal awake from the REM sleep, and also that the impair, and also suppress that the basic events happens. And as mentioned earlier, previously that the known REM regulating regions are located at the brainstem area, which is anatomically quite far from the medial prefrontal cortex located at the frontal part of the brain. So I hypothesized that there would be some brain regions which can mediate that this prefrontal control of the REM, prefrontal control of the REM sleep. To find the possible mediating regions, I studied that the anatomical projections from the MPFC to that the subcortical regions related to that the REM sleep regulation. And we found that the dense projections from the MPFC existing in the lateral hypothalamic area and the lateral dorsal tegmental area, or the periodactyl gray area, the PH area. And these brain regions that in the lateral hypothalamus, the MCH melanin concentrating hormones are known to be promote the REM sleep, whereas that the cholinergic neurons in the lateral dorsal tegmental area also seems to be promoting the REM sleep, whereas that in case of the PAG, the GABAergic neurons working as the REM suppressing neurons. To find that any candidate regions mediate the REM sleep regulation by the medial prefrontal cortex, I performed that the optogenetic stimulation of the MPFC projection to target optogenetic stimulation of the MPFC projections existing in the other brain regions. And in case of the stimulation, the stimulating projections to the lateral dorsal tegmental area or the PH area result in that there was no significant changes in the state transitions or the about the duration of the REM sleep or frequency of the basic theta. However, when I stimulate that the MPFC projections from the in the lateral hypothalamic area, it's showing that the activation can induce that the transition from the non-REM sleep to the REM sleep, and also elongate the duration of the REM sleep, and then we increase the frequency of the basic theta, which repeat that all the changes induced by the MPFC activation, which means that the lateral hypothalamus would be the brain region can mediate that the prefrontal control of the REM sleep. And to confirm that whether this MPFC to the lateral hypothalamus projections can really mediate the REM sleep regulation, I used the combination of the retrograde Cree and then the Cree-dependent channel C, so that injecting the retrograde Cree in the lateral hypothalamus and Cree-dependent channel C is injected in the MPFC. So only the MPFC neurons projecting to the lateral hypothalamus is stimulated during the sleep. And this specific activation of the MPFC subgroups showing that when the laser stimulation is applied, that is similar to the activation of the medial MPFC pyramidal neurons, that it can successfully induce that the non-REM to REM transition. So, which means that the activation of this lateral hypothalamus projecting MPFC neurons is sufficient to induce the non-REM to REM transitions, during the sleep. And also, closed loop stimulation of this lateral hypothalamus projecting MPFC neurons shows that that elongation of the REM sleep duration happens, and then the frequency of the basic theta is also increased, means that the lateral hypothalamus is the brain region can mediate the regulation of the REM sleep, and then the basic event by the medial prefrontal cortex. And in contrast to that, the activation experiment, optogenetic inhibition using the IC++ shows that IC++ shows that optogenetic inhibition of these lateral hypothalamus projecting neurons can suppress that the non-mental REM transitions happens. And the closer stimulation also showing that the duration of the REM sleep is reduced, and then the frequency of the basic theta is reduced, means that reduced means that also that lateral hypothalamus projecting neurons inhibition of the lateral hypothalamus projecting neurons work in the opposite direction of the activation. And to see that this effect of the inhibition on the sleep architecture of the lateral hypothalamus projecting MPS neurons in longer time scale, I used the chemogenetic inhibition method to inhibit the lateral hypothalamus projecting MPS neurons using the inhibitory dread virus combination of the retrograde 3 and then the inhibitory dread viruses. And similar to that, the optogenetic inhibition, long-term inhibition of these neurons reduce the amount of the REM sleep and then increase the amount of the non-REM sleep, and also reduce the frequency of the basic theta. I reduce the frequency of the basic theta in here. And to see how this lateral hypothalamus projecting MPS neurons behave during sleep, I proposed the cellular level of the calcium imaging using the microendoscope. And each line represents the activity of the cells circled by the same color on the field of the view. And then the consistent with the photophotometry, many cells became active during the REM sleep, but some cells also active during the wake, or some cells even during the non-activated during the non-REM sleep. And the traces of this calcium activity of this lateral hypothalamus projecting MPS neurons, showing that their activity becomes active in the different states per each cell. So I classify the recorded neurons according to its most active status. By average state activity, over the half of the lateral hypothalamus projecting MPS neurons are most active during the REM sleep, whereas that there are some cells active during the wake, most active during the wake, or they active during the non-REM sleep. And these REM active neurons can be further classified whether they are more active during wake, REM wake non-REM neurons, or they're more active during the non-REM sleep, REM non-REM wake neuron. And the next, to indicate the role of this REM active neurons, I analyzed the activity changes of these neurons at the point of the state transition. At the non-REM to REM transitions, that the REM non-awake neurons, which indicated by the blue line here, can show that their activity could start to increase earlier than the actual REM onset, means that this REM non-awake neurons seems to be involved in the induction of the REM. And then I also see that what is the role of the other type of the neurons, which is the REM wake non-REM neurons. Whether these neurons are involved in the maintenance of the REM sleep. That I divide the REM sleep with the REM sleep out with the threshold of the 30 seconds, and then compare the normalized activity of these two REM active subgroups during the short REM, shorter than the 30 seconds, and then the long REM sleep, the longer than the 30 seconds REM, the 30 seconds of the sleep. And this data showing that whereas that the REM non-awake neurons shows the similar activity regardless of the bout of the REM sleep duration, the REM wake non-neurons shows the significantly higher activity during the longer REM sleep, which means that this type of the neurons seems to be involved in that the maintenance of the REM sleep. And then they also related to that how these REM active neurons are behaved at the basic event during the REM sleep. I compared that the activity of these two different type of the REM active neurons at the point of the basic theta oscillation happens. And it turns out that whereas the REM non-awake neurons did not show significant different activity changes during the basic theta oscillation, but the REM wake non-neurons seems to be they show the significant increase at the point of the basic theta, which means that whereas the REM non-awake neuron seems to be more involved in the induction of the REM sleep, this REM wake non-neurons seems to be involved in that the elongated maintenance of the REM sleep and the basic event happening during the REM sleep. And as the final, that to see that what could be that the possible inputs to this lateral hypothalamus projecting MPF neurons involved in the regulation of the REM sleep are using the monosynaptic rabies tracing, and they found that this lateral hypothalamus projecting MPF neurons receive inputs from the various brain regions. These brain regions, including that the anterior cingulate cortex, and then the septum regions, that the ventral hippocampus and anterior thalamic region, and also interestingly that the medial septum region, which is involved in the regulation of theta oscillation during the REM sleep, also sending the quite large projections to the lateral hypothalamus projecting MPF neurons. So in conclusion, the activity of the MPF prefrontal neurons can induce and maintain the REM sleep, and the promotes the basic events like the rapid eye movement and then the basic theta oscillation. This regulation of the REM sleep is performed by the MPF cells projecting to the lateral hypothalamic area, and the majority of these lateral hypothalamic projecting MPF neurons are most active during the REM sleep, and the REM wake non-neurons shows that the activity increase and involved in the facilitation of the basic theta events happen. So in the introduction, I mentioned that the study about the REM sleep regulation circuits are mostly focused on the brainstem area and then the hypothalamus area, which is the posterior part of the brain. And the role of the cortex in the REM sleep regulation has not been identified much. But in my research shows that the MPFC has an ability to regulate the REM sleep and its basic features through its projections to the lateral hypothalamic area, which is actually involved in the functions like the reward, stress, or the memory, lateral hypothalamic area. So that considering that the MPFC and then the lateral hypothalamus are involved in the various functions, including the reward, stress, or the memory, my research implies that the underlying mechanism on how REM sleep and these cognitive or the physiological functions are connected. So for the future, that they try exploring that what could be that post-synaptic output of this MPFC neurons in the lateral hypothalamus would be that the interesting feature study which can be performed, because that the lateral hypothalamus is composed of the really various cell types, and then many of them are involved in the regulation of sleep and wake through the projections to the other brain area. And also considering that the lateral hypothalamus performs the other physiological functions and behaviors, identification of these postsynaptic neurons receiving MPFC projections may reveal that the relationship between sleep and then the other physiological behaviors. So with this study, that I got the large help from all the members of the web lab, the web lab, and then the Jung lab at the University of the Pennsylvania. Yeah, thank you for listening my presentation, and then I'm happy to answer any questions. Thank you.